Hello. So this will be my video on classical genetics. This video will take a little bit of time, and I'll go through some things quickly. But remember, you can pause, you can stop, you can rewind something, watch it again, do whatever you need to do to get the essential information. When we get into genetics, there's a lot of vocabulary terms that all seem pretty similar, so you want to keep track of them well. So let me share my screen here. Here we go. So I want to go back to something we've done this year, and that's the human karyotype. Remember that humans have 46 chromosomes. Most of them are the autosomes, which come in pairs. One from your mother, one from your father. And what's important to remember, if I can quickly annotate this, is that if we were to look for a gene, a sequence of nucleotides that code for a protein, we would find the same gene here as we do here. Essentially, you have two copies of every gene, a distinctive sequence of nucleotides, the order of which determines the order of amino acids in a protein. But that being said, it doesn't mean they're identical. Genes come in different forms, and we call those forms alleles. And that's going to be a key word. I'm going to say allele, dominant allele, recessive allele coming up. So they are versions of a gene being one distinctive sequence found in one particular location on one of the particular chromosomes. And this is the weird thing that's really hard for students because of the way our chromosomes align after fertilization. You have two copies of each gene. So we have kind of a redundant system built up that kind of play together to create who you are or what our pea plants are like that Gregor Mendel studied. So here's, let me just erase these lines quickly. So here's the man. So he's Gregor Mendel, famous for genetic experiments on pea plants in the late 1850s and early 1860s. He lived, worked, ran monastery. This was kind of his hobby. In his free time, he did some pea plant studying. He was an avid gardener. So not really a scientist, but kind of a hobby. But today he's known as the father of classical genetics. He kind of died not knowing how important his work would end up being. But today he's one of the most important people ever in genetics. Some important terms as we go through are genotype and phenotype. So your genotype is the alleles you have for a trait. So what the actual alleles are, and there's always going to be, remember, two of them for what we're going to do. But then that genotype creates what we call a phenotype, which is the actual appearance that comes from those genes or the interactions of those alleles. So genotype versus phenotype. In Mendelian genetics, we have a dominant allele and a recessive allele. So a dominant allele is an allele that is fully expressed when carried by only one homologous chromosome, while a recessive allele is an allele that is only expressed when carried by both homologous chromosomes. So dominant is, of course, dominant to the recessive. And this is kind of a Mendel term. He had what he called true breeding plants. True breeding plants were plants that always provided the same characteristic again and again. If he was interested in purple flowers, whenever he bred his purple flower plants together, they would always give him purple flowers and so on and so on and so on. He called them true breeding plants. Today we know those plants were the way they are because they either had two dominant alleles that gave the dominant trait, or two recessive alleles that gave the recessive trait. So two other important terms are homozygous and heterozygous. So here we can use our roots. Homo means same, so same alleles. So you're either homozygous dominant, two dominant alleles, or homozygous recessive, two recessive alleles. Notice we use an uppercase letter for the dominant allele and a lowercase letter for the recessive allele. Or you can be heterozygous, you can be different and have a dominant allele and a recessive allele. I'm going to do this on paper, but there are slides here that do the same thing for you. There are slides 6 and 7 on this slideshow that I'll post. But Mendel was famous for his generational studies that gave what he thought was kind of an odd result. So let me talk about the odd result, but I'll end up doing this on paper. He did this for all sorts of different characteristics found in pea plants and found this same ratio that we'll talk about popping up. So he bred what he called his pea generation. These were his true breeding plants that always provided purple flowers to purple flowers, white flowers to white flowers. And then he bred them together to create what he called the F1. These F1 plants all looked like one of the parents. In this case, purple flowers, no white flowers. But when he took his F1 generation and bred them with other F1 plants, something strange happened. He got purple flowers, but he also got white flowers. So white was able to go from one generation, skip, and go to another. So obviously there's something going on here that's hidden underneath the surface that Mendel was observing that he didn't understand, but we certainly do today by understanding the genes. So that's what I'll kind of take you through as we work out these problems. I also have steps for what these are called simple monohybrid crosses here, but you can watch my video and go through what I'm doing. So let me stop my screen sharing and switch to my document camera. So we won't look at flower color like in that example. We'll look at seed color, another trait that Mendel observed. And in my example, I'm going to have, again, a dominant allele and a recessive allele. Dominant allele will be a big Y, yellow, recessive, little y, green. There are 3 possible genotypes always in Mendelian genetics. You can be homozygous dominant, big Y, heterozygous, big Y, or homozygous recessive, little y. Note that homozygous dominant and heterozygous both provide the same phenotype, and that's yellow seeds because of the dominant allele that's being expressed. The only way to get green is to be homozygous recessive to show that recessive trait. That's important. 3 genotypes leading to only 2 possible phenotypes in Mendelian genetics. So let's start our example. And let's have, let's do a P generation to an F1 generation. That's what I'm going to end up doing. So I'm going to cross a homozygous dominant plant or a true breeding plant for yellow seeds with a homozygous recessive plant, little y, or true breeding for green. And see what happens. See what my offspring are like. So that's what Mendelian genetics is all about. It's about predicting offspring, what they're going to be like, how parents lead to certain characteristics in their children. And to do that, we use the famous Punnett square as a tool for us to understand what's going on here. So it's a 4-box setup where you're going to take the alleles of the parents and place them on the outside of the Punnett square. One parent on one side, one parent on the other. And then you're going to distribute them into your Punnett square. Distribute big Y's, distribute big Y's, distribute little y's. And notice in this one, each of my boxes is the same. Each of my possibilities for what an offspring could be like are the same. So I'm going to say 100% of my offspring are big Y, little y, or they're going to be heterozygous. This I would call the genotype. That's what the genes are of my offspring. I could also describe their phenotype, what they look like. In this case, since I only have one genotype, I can only have one phenotype, and I am 100% yellow seeds. Of course, Mendel didn't stop there. That's not very interesting. What got interesting is when he took these F1 plants that were heterozygous, bred them together to make his F2 generation. So place my Punnett square again. Now my parents are these heterozygous plants, big Y, little y, big Y, little y. Again, I distribute in. And we end up with a far more interesting result. Let's start by looking at what the genotypes are in this particular Punnett square. So I have one of my 4 boxes, 1/4 is a 25% chance of being big Y, or homozygous dominant. 50% chance of being big Y, little y, 1, 2 out of 4 heterozygous. And then to add up to 100%, a 25% chance, 1 out of 4, to be little y, or homozygous recessive. Of course, genotypes lead to phenotypes, what an organism actually looks like. And in this case, the homozygous dominant and the heterozygous plants are going to look the same. So I add up those percentages, and I end up with 75% yellow seeds and 25%, that's the only way to get the recessive character, is to be homozygous recessive, green seeds. And I completed my problem utilizing my Punnett squares. So that's kind of a start there. But to be successful in this, you probably have to do a couple of different things. You're going to have to use my video, use my PowerPoint. I'll also post another YouTube video about classical genetics that will help you solve these problems and get going on this process that you're not only going to use here, but eventually in the Wisconsin FAST Plant lab report that we're leading up to. Have a good day.